Okay, okay. Oh my God, I didn't want to scream, sorry. But maybe it wasn't enough. Um, we're ready to start. Okay. I'm sure there is a lot of discussion going on following the presentations. But now it's time for our last speaker, Carlos Ponce. Carlos Ponce conducts research at the intersection of visual neuroscience and machine learning with a focus on how different brain regions collaborate to process motion and recognize objects. His work employs a combination of electrophysiology, reversible deactivation, microstimulation, and computational modeling to explore these neural interactions. Ponce earned his MD PhD from Harvard Medical School and holds a Bachelor of Science from the University of Utah. His research has applications in automated visual recognition systems, including medical imaging and autonomous technologies. His talk is entitled On the Relationship Between Single Units and Distributed Representations. Thank you. And
all right, thank you very much. All right, well, thank you all for being here. Thank you, Karim, and the organizers of the conference. It's been a really, really great conference, and I can see how it sparked so many great collaborations across the globe. So thank you for the invitation. All right, so let me tell you a little bit about some problems that have been interesting, been of particular interest to me recently. Let's start with two simple facts. One simple fact is that pretty much every action, every thought, every movement that you make is the result of a series of neurons acting together in concert. You are a population of neurons. But at the same time, the other truth is that pretty much the most elemental unit of information transmission in the whole brain comes down to spikes, comes down to one neuron firing one spike to another neuron or not. And that to me becomes like this very interesting, almost paradoxical situation. It's like, how does all of this information transmission really break down to individual neurons chatting with each other? So one way in which I study this is in both ways. I study like at the level of the neurons and at the level of in biological networks. But also this is a question that we can address in deep networks of vision that are also artificial. So I study the visual system in the macaque. And I studied using electrophysiology at the level of the single unit and also at the level of microclusters, which you can think of as multi-units picked up by the electrode. So when it comes to vision, there's a couple of accounts that I like to always revisit when trying to explain what is it that, how is it that individual activity relates to population. And one specifically comes from Simon Thorpe's article, Local versus Distributed Coding. Here I would invite you to consider the activity of the neurons in a population as being part of a Cartesian coordinate system where you have neurons, every axis corresponds to the activity of one neuron. And so if you start with the idea that you have a concept, like a nice, interesting concept, very simple one, like a red square. In the localist code, you could imagine that every neuron is going to be able to encode a readily pre-named concept. So you're going to have a neuron, and every time the neuron fires a lot, that's because the red square is present in your visual field. The alternative to this, the far alternative, would be one where you have everything much more distributed. And so you have a red square, and that red square, that concept is now represented as part of a much more ambiguous set of activations over the whole population. Now, in this particular perspective, in a distributed code that is fully distributed, the idea is the single neuron is really going to carry very little interpretable information. And in a way, it makes one wonder, should we study the single neuron? This is something that I think has been kind of visualized really well and described really well in the idea of the neural population doctrine. The idea that you can think of the population, of the vector of activity, as a really elemental computational element. And that allows us to do a lot of really cool studies, like using clustering, representational similarity, or linear decoders to try and understand the brain, which is good. It's fine. And of course, the age of big data is sort of encouraging this perspective, right? Because now we're moving to the world where we can record from neuropixels, high-density probes, and now we can record from hundreds or thousands of electrodes, of like neurons at the same time, or many, many hundreds or thousands of voxels. We could even like look at individual hidden units in deep networks. So it just kind of becomes, tempting to think about just the population. So is the population the essential unit of information? And one thing that it does for me is that it creates a sort of a prediction. right? Because you can have two very different representational similarity spaces. And if you have two concepts, like a monkey and a cat, you can represent the distance between them in effectively a degenerate number of ways. You can have one brain that represents it like this, and then you can take the exact same number of neurons and completely rotate their encoding in such a way that they would be completely different in the way that they function at the single unit level. Is this true? I mean, so the fact is that the more, the last 70 years of recordings of electrophysiology in the brain kind of suggests that neurons do seem to rediscover similar kinds of tuning features and also values within those features, things like directions of motion, disparity values. It seems like you observe them from brain to brain. And in fact, even if we look at machine learning models that are just there to basically learn from the data, you could see that there are filters that are learned at the very first convolutional layer, which we can actually visualize because they're operating on RGB values. And you can see that a lot of these look very similar. And in fact, a lot of them look so similar as to even remind us of the work done by Hubel and Wiesel in the 1960s when they were actually mapping by hand the receptive fields of neurons in V1. And as you can see, this neuron that they mapped is also present like in different parts of these networks, like here and here, and another one right there. So it does seem like indeed there is some kind of anchoring of specific good solutions for neurons that might relate to the whole population. So how do we find them? What are these types? This is the kind of question that I'm interested in, why there are some neurotypes kind of reappearing. But to answer this question, we really have to answer the question, what are they? We need to understand the neuron a little bit more. So here's the simplest premise to the work that I'm going to tell you about. The premise is this. Visual neurons have receptive fields. That means there are regions in the visual space that will activate their responses. But those receptive fields also have shapes, meaning that while it's actually trivial to get a few spikes out of any visual neuron, just turn the lights on, you can do that. The fact is that if you can identify the specific shape within the receptive field that activates the neuron, you're going to get more spikes. This is the work that basically was illustrated by the work of Hubel and Wiesel in the 1960s. And their setup is basically still the best way that we can use to try and understand the visual neuron. It's quite simple. We introduce a microelectrode in visual cortex. In their case, it was primary visual cortex. We identified the receptive field, the area of where we're going to get a response. And then what they discovered was, in fact, that we can take any kind of image, which serves as the hypothesis of what specific neuron is encoding. And what they discovered is that V1 neurons were able to respond more to some specific orientations over others. They discovered orientation selectivity. This work, not just this work, but other kinds of work in deprivation, earned them the Nobel Prize. So it's a testament to their brilliance. But what's interesting about this particular result is the way that they actually discovered this. And many of you know this story that Hubel and Wiesel were basically studying visual cortex, and they were using an ophthalmoscope or a projector that worked on slides, and they were at first using this kind of visual stimuli, which they inherited from Steve Kufler, their mentor, to stimulate LGN. But they found that V1 neurons really weren't being activated as reliably as other cells. And as the way that Hubel says it is that our early failures were a matter of finding the right stimulus. So what they noticed is that as they inserted some of these slides, which had these painted dots on them, the neurons just kind of became activated. And they basically deduced that it was the moving shadow cast by the edge of the glass slide that was actually causing the responses of this neuron. So from the very beginning, trying to understand the single neuron combined not just brilliance, of which they had plenty, but also a little bit of luck. And that's the point. So how far can we push this luck, right, when we try to understand the rest of the brain? About the same time, we have Charlie Gross across the river, also in Cambridge, trying to understand how inferior temporal cortex neurons actually function in the brain. And like Hubel and Wiesel, they were making use of every kind of stimulus that they could, including things like their body parts, burn Q-tips, whatever they could find in the lab. And here the result that Charlie Gross described is that a lot of these cells were really selective for some kind of shape, texture, or color, but it was very difficult to specify what the unifying principle was between the responses of these neurons. So apparently the similar stimuli is the way that he phrased it. So what did he mean by that? Well, here's some recordings of some neurons in inferotemporal cortex that come from my lab. So here we have an inferotemporal cortex multi-unit, monkey B. And as you can see, this unit responds, this cluster responds pretty well to a lot of monkey parts. These are not all monkey parts, but perhaps you'll excuse me and say, this is a monkey neuron, okay? All right. But it gets harder for you to kind of cut me a break when it comes to these kind of neurons, right? Now, here's a neuron that's responding to a stop sign, to a bedroom, and to some kind of cat peering through a door. A place neuron, I don't know. But then this ones, and at this point, I just have to like throw up my hands and say, I have no idea what these neurons are encoding. Perhaps they are part of some kind of distributed code. Charlie Gross also said, you know, perhaps we never found the best stimulus for each one of these units. Here's the problem. Most of the neurons that you will encounter in the brain are like this. I like to think of these neurons as a sort of the dark matter of visual cortex because, A, most neurons, like dark matter in the universe, really this is what makes up most of these neurons, and especially in frontotemporal cortex. And I like to contrast this with the regular matter neurons that you probably have all heard about, the things like face neurons, which have been fantastically characterized by Margaret Livingstone, Doris Tsao, and many others. And these are neurons that really do align pretty well to some semantically interpretable features, and they can be found by using fMRI to localize specific patches that are become nicely activated, stick an electrode in them and record the responses, and it works really well. So they exist, but the fact is that they're not, they may not represent the entirety of what a visual cortex does. Nevertheless, the standard view has become where like most of the early visual neurons are thought to represent edges, like Kevin and Wiesel demonstrated, then something, 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 and then at the end you have object representation. And this is a part that I think I want to say is needs a little readjustment because when we record from these neurons, this is the kind of like presentation of the visual system that really emphasizes what is interpretable and really ignores a lot of the reality of what we see in visual cortex. So this is what I want to do in my research program. We basically want to create a framework of understanding that takes into account the semantically interpretable neurons, but also the heaps of the less interpretable ones. And what I want to make the case is that to really understand this, the formula is going to be the same. It's just a matter of how we prioritize it. How we're going to learn what visual cortex does at the level of neurons. In all of these scenarios, we're going to basically be using intuition, theory, and luck. But what I'm hoping we can do is actually operationalize luck into something more like discovery. And the question is, are we going to now take an intuition-first approach, as has been done quite well, but now maybe perhaps a discovery-first approach? And to do this discovery-first approach, we're going to basically get some inspiration from machine learning. We're going to basically take a lot of their tools and a little bit of their philosophy. Now here's something that will remind you of an earlier lecture today. The bitter lesson. Let's start with the philosophy of this. I'm not quite, as much as a devotee to Rich Sutton's bitter lesson as Owie, I believe. I don't know if he's here, but I do think a lot of inspiration from it. So you guys remember the bitter lesson? Show of hands. Most of you. Okay, great. Yeah, I love this blog. It's quite provocative. This idea that, as he argues, in artificial intelligence, building in how we think doesn't really work in the long run for scaling. So he says that AI researchers have often tried to build knowledge into their agents. This helps in the short term. It's personally satisfying to the researcher. But in the long run, it tends to plateau and even inhibit further progress. And he suggests that instead, breakthrough is going to come by a combination of search and learning. And while I don't quite agree with everything Rich Sutton says, I do believe that there is something interesting about the idea of using search and learning to kind of weaponize the part that is luck when it comes to visual cortex studies. And this time I'm going to tell you how we do this using generative networks. So, quick review. There's a lot of very useful models in visual neuroscience. You've seen them all today in some of the earlier lectures, so I don't need to go over them very much. But in all of these cases, these networks are very good at learning the visual statistics, the low-level statistics of training data like photographs. We have discriminative models, which are very good at taking an image and then providing an output of probabilities telling us what's in the image. And then we have generative models, which are, as you guys all know, sort of the inverse, where we can take a random noise vector and then train this model to basically map an image to one of these noise vectors. Now, specifically useful for our work has been the development of generative adversarial networks. which, as you know, are called this because they're trained under this circumstance where you have a network whose job is to learn, to kind of create natural images, and it's always pitted against a discriminator network whose job is to say if it's fake or real image. And if you get them to fight long enough and you get them to kind of train stably, eventually the generator becomes a very good model at kind of picking up some of the low-level statistics of this network, of the visual scenes, and kind of create new kinds of examples. These are two different kinds of networks. One's an older one, DeepSim, which we use a lot, and this is Big GAN. So the way that we try to use these statistics to kind of weaponize this idea of luck is using a closed-loop generative approach, where we can take some of these generators and just kind of couple them directly to the activity of neurons in a monkey's brain, and then kind of combine their information in such a way that a search algorithm is able to optimize and activate the neuron more and more. So I'll walk you through these steps one by one. So the very first thing that we do, that's still very old school and kind of an irreplaceable thing, is that we have to identify the spatial location of the receptive field, right? So what we do is we'll have the animal look at a dot, and then we just kind of flash images in different locations of the visual field. Monkey gets a reward, and that allows us to kind of localize the area of the receptive field more or less. It tends to be a bit of an estimate, but we get it. So then we're going to take the generator. It's going to create a bunch of random images, and next we present them to the animal. So this is all like in the way that's always been done, except that now we're going to present them to the animal. We're using chronic macroelectroarrays implanted in primary visual cortex, and before in frontotemporal cortex, and now more recently in prefrontal cortex as well. And I'm going to be showing you some results from a couple of monkeys here. And here's an example of basically what that looks like. So here's the animal. It's getting to his reward with the juice, basically, and we're monitoring the eye position. And now I'm just going to basically give you a little tour of what it's like to go to the lab. So we're listening to the spikes. That rhythm are the images kind of flashing at 100 milliseconds on, 150 milliseconds off, 3 to 5 stimulus per trial. And then we also interpose some natural images to kind of keep track of the stability of the recording, make sure that we're not losing isolation, and then changes in the firing rate could just be us fooling ourselves. The point is we record the responses to each individual neuron, from each individual neuron, and then we basically provide this information to the search algorithm. And the way that it works is you can think of the list of values that create every image as a kind of a chromosome. And so as you find that some of the chromosomes don't elicit a lot of activity and some do. And so what we do is we identify the ones that cause more activity, and then we just sample the parameter space around this one location. And then we create a new set of chromosomes, 30 or 40, give them back to the monkey, and then see what happens. So in a typical experiment, so here I'm showing you the original sets of images that we begin with, and some natural images that serve as reference. And as you can see for this particular neuron, this neuron responds quite poorly to this initial set of images. But then, during the experiment, the firing rate begins to climb and climb and climb until it begins to plateau, while at the same time, the responses to the natural images just kind of undergo a little bit of adaptation. So this, if you look at the change in the response over the whole experiment for the natural images on the x-axis and the synthetically changing images on the y-axis, you'll find that indeed this is a process that seems to work in pretty much every visual area with a receptive field that we can find, including now prefrontal cortex. Why does it work? What is it doing? Well, we've done a lot of investigations for it, but really the bulk of it is the image itself, right? So here what I'm going to show you is basically a 10, 15-minute experiment, just kind of compressed in just a few seconds. We're taking the average of all the frames, and basically it looks like this. So this is one neuron in V4 that is driving the synthesis of this one feature right there. A little black line surrounded by a lot of white space. Here's some individual frames. Not very surprising, I hope. This is exactly the kind of neuron that Hewlett and Wiesel had discovered, 60 years ago. Simply an orientation-tuned neuron. Here they are drawing it with their hands. We had to use artificial intelligence. They use their biological intelligence. That's why they're better than we are. But if you're cool with this, what we can now do is go and find every one of these single neurons and then just ask, what is it that they encode? So what you'll find, we like to refer to this neural guided image synthesis as prototypes just for the ease of discussion. And what you'll find is a lot of these prototypes are somewhat reminiscent of things that you would see in the natural world, but they are surely not semantically interpretable images, things that you might see in a face or things that you might find in a specific texture. So they are not interpretable, and I think that explains why a lot of these neurons seem to kind of be responding to this weird mix of natural images. We're seeing them like from a higher level perspective, whereas neurons are focusing at something even smaller. I wanted to also mention that this kind of image maximization approach has also been attempted by Puya, hopefully around here, in V4, and other groups have also managed to do this in their own way. But what's interesting to us here is that these representations may not be semantic, but it does make me think a lot of Paul's talk yesterday, and the idea that perhaps they are sort of more pragmatic representations, right? Like if you're trying to represent perhaps just a fragment of something that should make you turn like an eye. You don't need to represent the whole face in all of the details. All you need to do is kind of capture the idea of something like an eye in your periphery, and then you look at it. Something more pragmatic about it. But more importantly for us, it could be the case that indeed some of these features are kind of also contained within this larger texture. So one of the things that we decided to do is, can we actually try to localize the most important parts of the prototype? And this is an issue that really goes across all of visual neuroscience. If I tell you that I have a neuron that responds more to faces, I still can ask you, what part of the face is it that really the neuron is responding to? Is it the whole thing? Is it the eye? Is it the mouth? So to answer this question, we decided to take sort of a shuffling approach. And this is work that Antonio Montanaro started to do, where you can take an image and basically randomly select locations in the image and just shuffle the pixel values within that image itself, within that local patch, in such a way that you can keep track and keep constant the overall contrast and the overall luminance, but just completely abolish the local information within that. So we thought we would use it to kind of localize this prototype. So here's an example of an IT neuron that responds in creating, as you can tell, these two important local features. Now by eye, we can already know that the neuron cares about one of these two things because we published this. Neurons tend to create a white space around the things that they really care about. They hate crowding, so they just basically put stuff around it. But I can't really tell if it's one of these two. So one of the things that we decided to do is to then use this approach. We scrambled local features, created about 30 or 40 variants, and then presented them back to the monkey, into the neuron. And as you can see, this particular neuron responds quite comparably to images having a lot of these occlusions. But if you occlude a specific region, the firing rate just goes down to 0 or becomes suppressed. And here's the images that are more activating, and here's the images that are completely suppressive. And so that tells us that it's going to be this particular feature right there that the neuron really cares about and drives its activity. This kind of approach we've confirmed across multiple, many experiments. You can take the exact same image, and if you just hide a very important local feature, firing rate just completely goes away. So this is kind of evidence that there is indeed some kind of local specificity to the way that neurons are responding across natural images. This is also, for those of you who are biologists, an example of an experiment of necessity. I've demonstrated that the feature is important, but can we also demonstrate that the feature is sufficient? And so for that, what we can do is basically start by randomly selecting completely unrelated natural images out of the data set of like 50,000 of them. So in this particular case, it was this. Now we can take this local feature that we thought was important and basically graft it or transplant it onto these otherwise unrelated images and then ask, does the neuron respond as we think it should? And indeed, here's the responses of the neuron to to these images that lack the local feature, and the moment you transplant them, the firing rate goes up. Not necessarily all the way up to what it could be, but the point is, it does demonstrate that the local features do seem to be quite sufficient for increasing neuronal activity. So why do this? Why would a neuron really kind of focus at that level of granularity? And some of the times, you can answer this by looking at the relationship of some of these experiments that we see. For example, here's a V4 neuron that evolved this local feature. If you put it onto any kind of image, it'll increase its activity. But you also find that some of these images were already doing pretty well. right? This image right here was already, a randomly selected natural image was already pulling in a lot of spikes from this particular neuron. So what was it? Well, here's an image that actually improved a lot when you gave it that feature. And here's an image that just kind of improved a little bit. And as you can see, this local feature that the neuron cared about right at the edge of the face was already present in a lot of these images. So here's another example. This neuron actually was doing pretty well with the random image as well. And when you see why, you see that it's because that was a contour that was already present in this picture of a cow. And same thing, we just kind of keep seeing that the image only needs to bear a certain amount of similarity or a little bit of that local feature within that image to drive its activity higher compared to other things. So all of this kind of would suggest that indeed, these neurons, as has been alluded to earlier in this conference, the localist code doesn't really kind of hold for a lot of these individual neurons. It really does seem like it's more of a distributed code. It's more of a single neurons are kind of representing very local features within any one given object. But what we really want to point out is that a lot of these neurons are also not uninterpretable. They can, in fact, carry local regions and textures that are quite constant over time. Could we find a neuron that actually does represent something like a face? So we're looking for them, but a lot of them, certainly the method that we have does allow us in convolutional neural networks to kind of extract rather composite and elaborate representations from hidden units. So we're going to be looking for them, but I have a student, or she just graduated, Olivia, who decided that she was going to basically go all the way to prefrontal cortex and try this approach to them and see if, in fact, she could evolve things that are much more semantic. And surprisingly, the features that we could extract from some of these ventrolateral prefrontal cortex neurons were still quite simple, suggesting that perhaps this distributed representation goes all the way to the front, but we'll figure it out. All right, so in the little bit of time I have left, I really want to get at the answer of like, okay, so what then? Let's say that we finally were able to identify what drives a given neuron in some of these dark matter neurons, what activates them? How do we use that? And to me, the very first question that I wanted to answer is like, how does that relate to a distributed population code from the very beginning? And so here we decided to do a very simple experiment, okay? We're going to take these monkeys and we're going to present an image multiple times to create a vector of responses across V1, V4, and inferotemporal cortex. So that's going to give us a vector that we can average and figure out how it works with noise. And then what we're going to try to do is to see if we can relate the individual activity of every neuron relative to its prototype and see if we can actually find a similarity and at the same time try and reconstruct that image by taking these local components. And this is work that was started by Binshu Wang when they were in my lab and then picked up by Alireza. We basically took that closed loop approach that I described earlier. And all you need to know about it is that instead of just maximizing the activity of one neuron, all we're trying to do now is to basically replicate the pattern, the observed population pattern starting from scratch. We're asking what do we get back. So here's an example of one of these experiments. What I'm showing you here is the response of the synthetic image of the population to the synthetic image versus the target image. And here we're going to be doing kind of this evolution process to see if we can improve this objective function, basically reduce the error, the difference between the vectors. And as you can see, there is a bit of an improvement. It's not great. And in fact, quite reliably, we can get the neurons to sort of the population to begin to respond more closely, more similarly to the original pattern. And we can see an improvement. But as you'll see, that improvement doesn't really go to zero as we would hope it would. And we know that it's not the algorithm because if we do the exact same thing with convolutional neural networks, you find that indeed we can get much closer to zero. So we know that we have the statistical power to actually make this work. It's not working, so why? Well, not working as well. And part of the problem is the noise. Remember, these are images that we're presenting basically once for the whole population. And so what Alireza decided to do is to try and understand how well these reconstructions can work when we actually focus on, when we actually incorporate the noise. And so what is this error ceiling that we can observe in this task? And there's various alternatives. Alireza first thought, okay, what if we take just the individual distance between every population vector? And if you do that, there's a very large error that you see across the whole population, even larger than we get. So the other thing we can do is take all of the vectors and divide them into two halves, and then ask what is the correlation between each average. And that gives us a very narrow error distribution. And finally, what Alireza decided to do, which makes the most sense, is that we were going to ask a kind of a held out approach, where we're going to leave one trial out corresponding to one image presentation, and then average the rest of the trials and ask what is the distance between this one vector and the average vector. And that's more closely to what we did in the actual experiments. And as you can tell, when we do that, we find that indeed these reconstructions were pretty much as good as possible, given the error that we get, the variability that we get when we present these images just once. So this happens across multiple monkeys, and now comes the part that is interesting, because we can ask, well, what happens to the images? And what happens is that we don't really get the images back. We get like local features back, but not the whole thing. And so one question that we have is, why are we not getting the full image back as we thought we could? A couple of hypotheses is that indeed this visual code perhaps is a little more emergent. Perhaps there's some kind of non-linear dependencies between the individual neurons that we haven't accounted for. Or it's also possible that the visual code is reducible, and yeah, maybe this does come down to those local regions that we should be paying attention to. Yes, please. Oh, almost out of time? Oh, gosh. Oh, gosh. I mean, how much? Oh, my God. Boy, I'm having fun. Well, I guess all I can really say is that basically if you take this approach and you figure out what the neuron is doing, how it responds across a lot of these images, they may see no relationship, but even at the level of the vector, we do see that indeed they respond to these local features. So these are images, natural images in the reconstruction that elicit a lot of responses from these neurons, and these are the ones that don't. And as you can see, the local feature is missing. So we can actually quantify the image distance between these specific image reconstructions, and we can find that there is indeed a correlation, a negative correlation between how different the images are and the level of activity that we get from individual neurons. And so that correlation can be quantified across the entire network. And so what it all basically boils down to is that these reconstructions can recover the local patch, but not the whole configuration. And that's part of the issue why I believe we're not seeing that reconstruction back. Yes? Another what?
It was an error that I made in calculating the time. So you have another 10 minutes.
I don't need those 10 minutes now. But now we can ask you questions. But anyway, I guess I'll stretch out the more pontificating part about what I think you should be the takeaway from this. But indeed, it does seem that really if we want to solve this issue of visual coding, the visual population code, we really kind of have to focus both at the local and population level. We really have to answer the question of what does the neuron do? And I think what we're trying to see is that a lot of the confusion in the field came down to the fact that we're trying to see the world, and we're trying to see images the way that some of these neurons, not exactly in the same way that the neurons see it. The neurons tend to be focused on local features a little bit more than kind of semantic or configurational concepts. But so far, the evidence seems to be that indeed a lot of this activity can be reduced to the tuning of individual neurons. And perhaps there isn't. We'll have to quantify how much there is emergence when it comes to the visual population code. There's something else that really I think about when it comes to this bitter lesson, right? This idea that as much as I really like what's been happening in the field, I feel like we got a little stuck in a rut in terms of what neurons are we thought neurons are representing. And yeah, part of the issue is that the view of the single neuron can be really hard for us to understand. Where we see a set of random images, and neurons kind of find a much more local, subtle similarity. And to me, I feel like then the idea of kind of front-loading interpretability it can be a bit of a problem. I think about it specifically when it comes to adversarial patterns. And I think in this conference, everybody knows what an adversarial attack is, right? But yeah, just to clarify, the idea is that, yeah, if you take a network that responds and correctly classifies this as a panda, we can find some pattern that we can superimpose on it in such a way that this network robustly, confidently is going to say it's a monkey, a gibbon, I guess an ape. And what's interesting to me is that these adversarial patterns are often treated to be problems. They're thought to be like, they're evidence that there's something wrong with convolutional networks and we should fix them to make them more like we see things. And really the whole point of an adversarial problem pattern being a problem is it doesn't make an intuitive sense. It's sort of like the folk psychology way, right? And so that's why I'm fascinated by this idea of results like by the Madri group showing that you can actually take some of these adversarial patterns, train neural networks just on the adversarial patterns, test them on natural regular images, and the networks do well. The networks do better than you would expect. And that tells us that it's not a problem with the network, it's rather these networks, and presumably visual neurons, are seeing things in images that are part of images. Like some of these patterns really are present in frogs, and they really are present in cats. We don't see them, but neurons do, and convolutional networks do. And part of the reason that I think that's also interesting is that if you try to make the neuron or the network more aligned to our perception, it gets worse. Like the classification accuracy, a lot of these networks actually suffers when you take away these local features that they seem to be paying more attention to. And I think that's interesting because I spent a little time in pathology. And in pathology, as you know, the job is to basically look at a piece of tissue that cannot be described by any kind of word that you'll find in a regular colloquial conversation. But the job of the pathologist is to make that pattern meaningful, to basically come up with words and descriptions that actually will tell the surgeon whether it's atypical, whether it's a problem, whether it's something that should lead to a surgery or to chemo or to nothing. And if that pathologist basically said, you know, this pattern is not human interpretable, the pathologist would be asked to leave the room, probably sued for malpractice, as he should. The point is that as we try and understand the neuron, as we try to understand the local workings of a neural network, we really have to not front load what we think, how we think the network should be. We really should just describe how it works. And I haven't thought of a good way to put this. So the way that I think about it is that we should be prepared really to kind of be ready to name what we discover when it comes to how the neurons work, and not just limit ourselves to discover that which we can already name. So, yeah, anyway, that's all I really wanted to tell you guys. Thank you very much for your time and attention, and I'll be happy to answer any questions.
Okay, 0 time for questions. I'm joking. Okay. Go ahead.
So, yeah, thanks for the talk. It left me wondering, how do you think the coding along the occipital infratemporal cortex progression, so to speak, what changes? And
Yeah.
yeah, actually, just before that also, are some of the features that you're finding something that a lot of people had found like a few decades ago in terms of like these little texture patterns? I remember these papers with these, like, I don't know, like these different kinds of patterns and unnameable kinds of things too. Is that
Absolutely.
Does that ring a bell? Or?
Exactly, Kobataki and Tanaka. Here's some of my favorite experiments done in visual neuroscience, which also answer your first question. The way they did this is they basically take a table with toys and objects, they show them to the monkey, then they find a neuron that responds to one of the objects, then they take a paper cutout and they begin to slowly strip away features out of that object, and they find that the neurons are very happy to respond. This is work done in the 90s, and it demonstrated that a lot of these neurons that are responding to objects were in fact responding to much simpler features. They didn't really fully characterize all of them, but then that to me seemed evidence that indeed what happens when you go down the ventral pathway, it's not really encoding for semantic concepts like an object, but rather to kind of create discovery of niche features. So these are features that are simple, just more likely to be correlated with things like faces, like a dot surrounded by a little curve. Mm-hmm. That allows you to respond to faces pretty well, but also it explains why some of these neurons will respond to things like clocks. And so I think that's what's really happening is that the visual system is getting smarter to finding those local features, but still allowing for distributed representations.
So you don't find something like that selective for faces?
Well, I think features contained within faces. Yeah.
I see. So only the features.
Yeah, I mean, but you know, I can't speak for the whole brain. I haven't studied all the brain. I don't, but right now, what we're starting to see, and I think that would seem to be the surest bet for us to do. Do I call it?
Go ahead. So we do one and one.
Oh, sure, thanks. So when looking at the PFC neuron, which is receiving inputs from V4, how do you know you're not getting stuck in a local minimum for a V4 neuron that's an input to the PFC neuron? Does that make sense? So like the PFC neuron is still going to fire in response to its input from V4, even if it's not getting all of the other inputs that might combine to a semantically meaningful object.
I see.
Yeah, but maybe you haven't found the object I'm not a proponent of the grandmother cell theory. I don't think we should be searching for this, but maybe there might be some sort of semantic information that could be encoded in these neurons or populations of these neurons, but you're not finding it. You're just finding inputs from V4.
Yeah, I think that's a perfectly reasonable possibility. One of the things we also did is we didn't train the animals to perform any task, and perhaps if we did, well, over time, we'd find that neuron that encodes for something simple then acquires a more complex, interesting representation. So yeah, perhaps what we're really doing is optimizing some earlier input to PFC, and then it could do something more interesting. I don't think it negated the fact that we could have found something else. We certainly had the technology. And the potential, but we didn't see it, so we'll report that. But I think it's possible.
Thank you.
Thanks a lot. Very cool stuff. I was wondering, like, if you do this evolutionary search, like for finding your local feature, does each cell only have one local feature, or like if you would reinitialize your evolutionary search with different random seed, that you would converge to a different local feature?
Yeah, so great question. So we've been recording for about a year from some of these same channels, and basically what I can tell you is that if you take an example of a channel that responds to a local dot, a dot that surrounds in white, black dot surrounded by white, if we evolve from that from multiple random seeds, we'll get the dot back. It can change its position a little bit. Sometimes we'll get 2 dots, occasionally 3 dots. And I think that tells us a little bit about the invariance that this neuron is happy to go for. But we never get something where like suddenly turns into something completely different, like a red square or anything like that.
But so that would be more like a local search around the feature that you then already got. Like I would be interested, like if you really initialize your evolutionary search from scratch, like whether you would actually converge to the same local feature.
No. So we do start from scratch every time. We start from the center of the latent space, completely different locations. And what happens is that basically the generator represents the same feature in multiple places. So basically, if you look at the trajectory in the latent space, we go to different places, but the feature is visually the same.
Okay, that's super cool. Thanks.
Yep.
Yes. Hi.
So with your reconstructions, I really love the idea. Do you think that perhaps your sampling is not distributed enough so that you could capture the diversity of features that are required to properly reconstruct the scenes that you...
Yes. So you mean like the spatial sampling cortex?
Yeah, more channels covering more of the cortex to make sure you get all the features.
Absolutely. No, I believe that that'll be true, and especially with neuropixels, as we were able to sample, that's because the fact is the brain does represent objects, right? It does represent texture. It represents absolutely everything. So yes, I think, but really when it comes down to what is the role of the individual neuron, I think what we'll find is that the distributed representation is the answer, and that will allow us to reconstruct. So yeah, I would agree.
Thank you so much for your talk. You talked briefly about adversarial decisions and how they can be features. Yeah. And it made me think about a little bit of how a child makes mistakes and how they're corrected or how they're helped by their teachers or parents. And I was wondering, first of all, what do you think about this comparison? And second of all, could we maybe use this in a certain way to help improve our algorithms in the future?
Interesting. So you're thinking about how a child learns, and you think that how would that relate to the adversarial patterns?
Yeah. So for example, thinking about verbal learning. So with the exceptions or irregular verbs, maybe a child would make a mistake on this verb, and then a parent or a teacher would say, oh, I get why you made this mistakes. This part looks like, or this kind of correcting or using those useful attributes.
Yes.
And then say, okay, but this is specific, or this is a new rule maybe
I
that
see.
you don't know. And yeah, so.
Yeah, interesting. Like, like my daughter, 5 years old, still makes a mistake of saying, like, instead of saying I broke something, she'll say I break it something. You know, she's kind of taking that local feature, putting it where it should go, and I say, I'm sorry, no, we say broke it, and it's a completely arbitrary thing. So yes, I mean, it does make me wonder if perhaps during education, as we train, as we grow up, we sort of have a tendency to see things in a more gestalt, configurational kind of way, but that could be partially learned based on some of these outside exercises. So yeah, I think that would make a lot of sense. But I am interested about the idea that a lot of our perception really is more fragmented, at least for the first 100 milliseconds that we think it is.
Thank you.
Thank you.
Okay. Make the question as quick as possible so that the other speakers are the happiest as possible.
All right, I'll try to. Brilliant talk. Thank you so much. I was just wondering, how much do you think that your conclusions are constrained by the type of network that you use? And
Yes.
let's say that you want to like explore the semantic space, just like dumb it down to just a few dimensions and just move across stuff that are semantically interpretable.
Yeah, right.
Do you think you would get something out of it at all?
So that's a great question, and stay tuned. So we did do this whole approach using a whole new network called BigGAN. And BigGAN is very good at creating objects. Everything in it is semantically interpretable. And one of the things that we discovered was that if you have these two networks fight each other to sort of make the neuron happy by activating it, especially in IT, you'll get to the point that the neuron is equally happy by both kinds of activations. One corresponding to a semantically interpretable thing like a dog, and the other one being a feature contained within the dog. And so that to us is evidence that indeed, again, the neuron is kind of seeing the local feature, but at the same time says, I'll take it if it's in a dog. Now, there are some neurons that do seem a little bit more excited by BigGAN than they are by this other network, and that's one of our publications. But I think to us the takeaway is, yeah, the local features still matter, but yeah, we haven't quite gone on to say it's the only thing that matters. That's a good question.
Okay. Thank you so much.
Thank you all.